This one.
All right, everyone. Welcome
Does
back.
that work?
We are now about to hear from Sean Brennan. He is the last of the early career speakers. I was really excited when I learned that the theme this year was isotopes, and I was on the committee because I knew we could get, or I hoped we could get Sean to come. Sean hails from a pretty long lineage of isotope nerdness. He was an undergrad at University of Utah with Jim and Ben, literally cutting his teeth because he was actually cutting Ben's teeth and grinding them for him. Well, not Ben's teeth, they were goat's teeth, whatever. Then he went on to Alaska Fairbanks to do his PhD, where he was using strontium isotopes to understand migration and production in salmon populations, which he will be talking about today. And then he's been continuing that work with Daniel Schindler at the University of Washington, which is where I met him, where he's now a postdoc, where he's been using newer techniques and sort of time series to sort of scale these ideas up. And just a little bit about Sean. It's kind of interesting. I was asking him last night, I was like, so how do you define yourself? And he was like, ecologist? And I was like, what about biogeochemist? And he was like, okay, biogeochemist. And then I said, what's your sort of tagline? And he had it ready, so he's obviously been applying for jobs. He says, how animals respond to heterogeneous landscapes and what that means for biological production. And I was like, oh, you're an ecosystem ecologist. And he was like, sure. And so the point of that being that what I really like about Sean's work is that he's integrating a bunch of different subdisciplines within our field, and particularly the things that I'm interested in, he's applying it towards thinking about conservation of some really important species. So without further ado, I'd like to introduce Sean. Sir.
Can everybody hear me? Okay, good. Thank you, Jake. Appreciate that. So I'm really excited to be here. Thanks for the invitation. It's an honor to get the chance to actually contribute to this symposium. I think what you guys are doing right now is really cool in terms of how you've organized it and set aside a specific symposium for fledgling scientists like myself. Okay, so my talk is going to be about how we can use isotopic variation, specifically strontium isotopic variation across landscapes, to gain insight into this general phenomenon in nature that's oftentimes called the shifting habitat mosaic, and how these types of dynamics influence the total biological production from landscapes. And I'm going to be looking at specifically fish production from these large, complex river basins. So this idea of a shifting habitat mosaic was laid out, at least for river ecosystems, by Jack Stanford and others. And essentially what they showed was that we can think about the habitat of all organisms across the landscape as really being the result of how local geomorphic features, things like topography, filter the overriding environmental forcing of a region, so things like the regional climate. And what's important about this concept is that it emphasizes the dynamic nature between these two. So you can have a large degree of environmental variation in a given region, but how that's actually expressed across the landscape, you'd expect that the spatial configuration of environmental conditions to actually change through time. And this has important implications for biological communities because over daily and millennial and perhaps even longer, like we've seen today, longer timescales, animals must be able to adapt and respond to these shifting patterns. And so a quintessential example of a shifting habitat mosaic is that of a floodplain. So here's an example of the Nyack floodplain in Montana. And what these workers showed was that the frequency and the magnitude of floods across this floodplain track the Pacific Decadal Oscillation. And over 60 years, this channel is migrating across the floodplain. And what it sets up is this mosaic of habitats over here that are all in different successional stages. There's places on the landscape that are frequently disturbed and those that are less frequently disturbed. And this has important implications for the fish communities, the invertebrate communities, because this mosaic influences things like wood recruitment into the stream. It influences how temperature is distributed across the floodplain, which are all biologically important parameters. So since a lot of work has started to focus on how biological communities actually respond to these. So here's a great example of how juvenile coho salmon actually track the shifting pattern of temperatures across this delta of a small stream draining into a lake. And depending on the lake level, the spatial configuration of the warm and the cold patches actually shifts around, and the coho track this to optimize both their foraging opportunities and their metabolic opportunities. And it's not these dynamics are not limited to aquatic systems. They play out in terrestrial ecosystems, and there's a rich literature there as well. So here's an example of how mule deer track the spring green-up as it propagates up an elevation gradient. And by doing so, they essentially extend the total duration of time that they have this high-quality forage available to them over the course of a calendar year. So that's two examples playing out at daily and seasonal timescales and at relatively small spatial scales. And it's not really known, or we have a weak understanding of how these dynamics play out across a range of scales, and in particular how they influence the total biological production from a system. For instance, fisheries. So we've been looking at these two questions in the salmon ecosystems of Western Alaska, these three large systems here, Yukon, Kuskokwim, and Nushagak. And for those of you not familiar with the salmon life history, they hatch in the freshwater system. They spend a certain amount of time there. They move to the ocean. They spend a certain amount of time there. They get fat and juicy. And then they migrate back, precisely back to their natal origin site, where they spawn and die. And this occurs over generation after generation. And essentially what happens is that it builds this locally adapted population structure that's filling out this large degree of habitat complexity that is composed and encompassed within each of these large river systems. And in terms of worldwide production, more than 50% of the world's wild sockeye salmon come from Bristol Bay alone. And more than 50% of the world's wild Chinook salmon production come from these three systems. And they support both commercially and subsistence fisheries that are critical to the regional and local communities throughout the region. So today I'm going to show how we can use isotopic variation across these landscapes to actually tease out how production of salmon actually shifts across these basins through time, but also how individuals and populations distribute the total amount of growth they need in the freshwater system before they migrate to the ocean. So it's important to point out that the production dynamics of these large rivers in terms of the total number of fish returning each year is essentially the result of an almost innumerable number of effects and associated population responses that are playing out across a really wide range of scales. And so developing any sort of way to understand these or to monitor these is inherently difficult and oftentimes intractable because these are huge systems, they're remote, this is a highly mobile species or set of species, and they experience really high juvenile mortalities. That's what the production pattern looks like for each of those 3 years in terms of the total number of fish returning. So our approach to this problem is essentially use strontium isotopic variation across these systems. And the primary reason why we use strontium isotope ratios is because what gets recorded in the otolith of fish, the ear stone of fish, directly corresponds to what the fish is swimming in. So there's a one-to-one correspondence. Here's 4 different examples, different species, different systems. It's a very consistent and very predictable relationship. The other reason is that the variation of this isotope ratio varies more or less predictably with geologic heterogeneity. And so you can have both large amounts of isotopic variation at both local scales and regional scales. And so given this reliable tracer, you then need an actual reliable recorder in each individual. And the otolith is a very good recorder. It grows via these concentric rings of calcium carbonate. And indeed, all fish have them, and here are some examples. And so what we do is we measure the strontium isotopic ratio from the beginning of the fish's life all the way through its freshwater growth or freshwater residence period before they migrate to the ocean. Now, the next piece of the puzzle is you actually need some sort of baseline across these big systems to actually compare what you're measuring the otolith to, and oftentimes they look like this. They're geographically incomplete. And so we use a set of spatial statistical models that essentially capture the variation that's being driven in isotope ratios in rivers as a function of the known drivers, things like geologic heterogeneity, plus the fact that locations within the network are spatially dependent, not in terms of just how the crow flies, but also in terms of how water is actually routed through the network downstream. And so with these models, we can then make an actual prediction of expected isotope ratios across the entire spatial domain in terms of the locations that could have produced a fish or were the habitat of a given fish. So we've done this for the Nushagak, we've done it for the Kuskokwim, and we've done it for the Yukon. And to use this information, we use what's oftentimes called a geographically continuous approach to this. And essentially what you do is you take your estimate of isotope ratios across all locations that could have produced a fish, plus the fact that you know what's in the water gets directly recorded in the otolith, and then you use conditional probabilities to say what are the most likely locations given isotope ratio measured in an otolith and that which you've predicted across the landscape. So I'm going to show you a few examples of what this actually looks like. So these are from Kuskokwim River. These are fish that are caught at the mouth of the river on their way back up to some unknown location within the vast Kuskokwim River. And based on, say, that otolith measurement there of fish 191, you essentially shrink the universe of all potential locations down to a single tributary within this really vast network. And as you measure different isotope ratios in different fish, those locations change across the watershed. So we've shown that this is very accurate and a very precise way of doing this, but what we're really interested in is actually looking at production patterns. So the way we do that is we take all of these probability surfaces of hundreds of individuals that have returned to the river for a given year, and we aggregate them all on top of each other, and we produce a map of the relative production of, say, Chinook salmon across these large river basins. So here's the production from the Kuskokwim River, and the key take-home here is that the production is not evenly distributed across the basin, right? There's places on the landscape that are highly productive and those that are less so. So with that, I'm going to shift over to the Nushagak River to look at how we can actually quantify this phenomenon, shifting habitat mosaics. So the Nushagak drains into Bristol Bay. It's the biggest of the watersheds that do, and it's unique in the region because in addition to these large, iconic lakes that produce a ton of sockeye salmon like the other watersheds, the majority of its land surface area is actually riverine habitat, some of which is not glacially influenced and some of which is heavily glacially influenced, plus this lower river region. And it produces a lot of sockeye salmon, and it produces a lot of Chinook salmon. Furthermore, in addition to those general habitat features on the landscape, if you look at the spatial variability of some of the usual suspects of environmental conditions that influence biologically important parameters, things like stream temperature and stream flow, three of which are shown here: snow cover throughout the year, precipitation amount, air temperature. These vary at multiple spatial scales, in particular these large, intermediate, regional spatial scales. So given the size of the system, the complexity of the system, you'd expect that the production patterns of the system should actually shift through time. And we can quantify that, like I just showed you, by using strontium isotopic variation across the watersheds. So here's two isoscapes, one for Chinook, one for sockeye. The only difference there is that the lake habitats are considered in the sockeye isoscape because they produce a lot of sockeye, whereas the lakes tend not to produce Chinook. So I'm going to first show you the data from the Chinook salmon. We've got multiple years. I'm just going to show you two of them. So similar to the Kuskokwim, you can see that the production, so those dark red lines are where places that are really productive, and the white lines are places that are less productive. And what you see, like the Kuskokwim, is that it's not evenly distributed across the basin, right? So you could look at this if you had 1 year data, and you could try to convince yourself that that part of the watershed, this part that's really red, should receive most of the conservation effort or attention, right? It's producing most of the fish. If we wanted to put a mine, say right there, like Pebble Mine, you could convince yourself that, you know, most of the production is coming from over there. But if you flip to the next year of data, the production shifts to the other side of the watershed. So I'm just going to do that again because I spent so much time on it. Okay, so the production is shifting across different areas. And we see similar things playing out with the sockeye salmon. So if we look at sockeye, again, we can see it's not evenly distributed, and it's concentrated in these lake habitats in this first year of data that we've got. You flip to the next year of data, and there's a big shift in where these fish are coming from, and it's much more evenly distributed with the riverine habitats. Now, what's interesting with the sockeye is that if you look at the production patterns of different well-established life histories of these fish, so those sockeye that spend less than a year in freshwater, oftentimes referred to as stream or ocean type sockeye, they tend to be more associated with riverine habitats. And indeed, if you look at the production patterns for just those fish, they are the riverine habitats are much more important, especially when you compare it to the same year of production, but for those sockeye salmon that spend at least a year in the system, which are typically associated with lakes. So if you take a step back and you think about, I just showed you that there's this complex interplay between the habitat diversity across the system, the population structure across the system, and this life history, locally adapted life history that is influencing how production changes year to year. But everything I just showed you was capturing the information in just the natal origin region of the otolith. And because of the way the otolith grows, we actually have this entire freshwater residence period that is stored that we can reconstruct based on the isotopes where these fish were throughout that period. And we've got this before they moved to the ocean, I should say. And we've got this for hundreds of individuals for each return year. So each of these lines is color-coded on the basis of their natal origin. So if you see a colored line track off into a different color band, that's evidence of an individual that used multiple habitats to achieve the total amount of growth needed before it moved to the ocean. And so the way that we quantify this is we came up with we developed this modeling framework we're calling space-time isotope models of provenance, where we leverage the spatial variation across the system, plus the fact that otoliths represent the serial record of isotope ratios, and then we generate essentially a provenance time series for each individual fish. And that looks like this. So here's an example of one of these. This is the otolith trace, and this vertical bar is going to go this way as this movie plays. This particular individual originated in the Mulchatna River. Before it moved to the ocean, it moved to the lower river, put on a substantial amount of growth, and then it moved to the ocean. This is cut off at the point at which the otolith is no longer freshwater totally, and this is the global marine value. Now, these models incorporate multiple sources of information and data. They also account for multiple variance-generating processes that are spatially and temporally explicit. And we did this for 1,400 fish for different years and different species. And so what's interesting is that you can use these models to then start to ask and look at these questions such as, well, like I just showed you, to what extent is that life history actually used? Is it an important life history? To what extent is the lower river just a migration corridor? And so what I'm showing here is the proportion of growth by weight that fish put on only in the lower river as a function of where they originated. And the gray bar here happens to be the range of ratios in the lower river, which is the most isotopically unique place in the Nushagak, just happens to be there. And the black line is the expected proportion of growth that a fish would put on if it originated somewhere, say over here, but it stayed there its entire freshwater residence. So if you see dots that plot outside of that gray box and above that black line, that's a fish that put on substantial amounts of growth in the lower river, but did not originate there. And what's interesting is these different colors here are different years, and the extent to which the lower river is used actually changes year to year. So here's 3 years of the Chinook salmon data. Some years it's used to a greater extent than others. Furthermore, if you look at the sockeye, we see some other interesting things. So this is the same plot, but for sockeye salmon. And what we find is that there's this interesting difference between these freshwater life histories. So those fish that are associated with riverine habitats, typically, these freshwater zero fish, tend to use the lower river to a greater extent than those fish that are associated with lake habitats. And so again, what we're seeing is this complex interplay between habitat diversity and these locally adapted life histories that's influencing how production is changing through time. So the other important question we wanted to look at was how these actually shift across spatial scales, how these shifts play out across spatial scales. And so what we did is we took those production maps and we aggregated them on the basis of stream order, which reflects this nested spatial structure of rivers, right? Low orders are these headwater streams. High orders are these main stem river environments. And when you do that, you can essentially look at the interannual variability in production by comparing these multiple years at these fine spatial scales relative to these much more aggregated spatial scales. And what you see for both sockeye and for Chinook is that at these fine spatial scales, there's a lot more interannual variability. As you aggregate to the production of the entire basin, that dampens. But what's interesting is that the variability in production is actually maintained across each spatial scale, which suggests that each of these scales is actually contributing to the overall dampening that you see at the entire landscape scale. Furthermore, when you compare the isotope-based estimates to an estimate where you assume that production is randomly distributed across the basin and then you aggregate them in the same way, which are those dotted lines here, right? So this is what you'd expect if production was randomly distributed across the basin. You can see that there's this increased variability at these intermediate scales, which suggests that there's some sort of non-random forcing going on at this intermediate scale that is influencing the production dynamics of the Nushagak. And when you compare what the spatial variability is in terms of these usual suspects of environmental conditions, there's a pretty good correspondence between the patch size of the largest amount of variability on the landscape for things like air temperature, precipitation, and snow cover, which further suggests that the shifting habitat mosaic dynamics that we've been discussing are influencing how production is actually distributed across these basins and how it shifts through time. And so, just to sum up, currently we lack an understanding of why we're seeing complementary production dynamics across different regions. But we are showing is that these intact free-flowing river basins are expressing shifting habitat mosaic dynamics across a large range of scales and dimensions of biological diversity, which ultimately stabilize the production of fish at the scale of the entire basin. And so, given that the most productive locations or those that are most suitable for growth are actually shifting across space and through time, this means that the stability of biological production across an entire landscape is more stable at aggregate spatial scales, which basically means that the conservation of the processes that generate and maintain this across these landscapes is just as important as the biological communities that they support. And so with that, I just want to acknowledge a very exceptional cast of mentors, colleagues, and co-authors that have all contributed immensely, not only to my own career as a scientist, but also to the work that I just showed you. In particular, Daniel Schindler and Turi Serling and Diego Fernandez have been very instrumental in getting this work up and going and producing what I just showed. I'm not sure who drove or who flew the helicopter that day. I'm pretty sure it was Diego because Turi was probably just taking pictures. But anyways, and I also want to acknowledge Tim Klein, who's now a postdoc here. He's been instrumental in getting some of this work going as well. And then this is work that is really field-based, model-based, and lab-based, and there's a lot of people that have contributed immensely to this. And then to the folks that have actually funded the work, I'd like to acknowledge them as well. So thank you, and I look forward to some questions.
How much of the variance across
space in the production that you see from year to year, do you think is due to strong cohorts of fish production? So, they return from the ocean, a large cohort comes up, and so you see much greater production in one part of the basin that year, and then the next year a different cohort comes up, and so forth.
Yeah, so there is a big part of the picture there is the fact that these returns are made up of fish that are of different age classes. So when some of the fish that are returning in terms of this aggregate production are going to be fish that spent 3 years in the ocean or 4 years in the ocean. And so in terms of, yeah, I mean, that's a big piece of the puzzle in terms of what drives. There could be a really strong cohort that exhibited a particular life history or experienced the right conditions at the right time that is influencing this production. And right now, we don't have a way of actually looking at that. But what we're showing is how these different parts of the basin are flickering on and off through time. But on that note, there is a strong, so I have some backup slides. There's a strong link between the freshwater habitat and marine survival, which is related to this. And so the first few months in the ocean, when these fish actually, this is from the Fraser River, when these fish actually hit the ocean, that is a big determinant in terms of the success of that given year of migrating salmon. And depending on if these fish migrated early or they migrate late, they may miss or hit right on the money the optimal conditions in the ocean, which would set the strength of this habitat component versus this habitat component in terms of when these fish actually migrated. So it's an integral piece of the picture in terms of the phenology of these different populations, the strength of the cohorts, and how that plays out is an important piece of the picture. Yeah. Yes. Go ahead.
Is the water temperature in the rivers and streams is always in a suitable range, or does that drive the movement, spatial movement of the?
In the Nushagak, I presume we don't have an extensive temperature measurement database across this really large region, but given where this place is on Earth, I presume it's not reaching the 25-degree Celsius threshold that a salmon would take a nosedive. Yeah.
So along those lines, this is more of an ecology question, and you kind of hinted at that, but what is a salmon like? Like what is good salmon habitat? What's the ideal sort of combination of conditions that would maximize production?
Yeah, so that's a good question. That is the suitability of habitat, it could be a combination of a whole range of things. It could be physical, it could be chemical, it could be biological. And we don't have a huge suite of things that we've measured across the basin to do that. What we simply asked was, well, the production is going to be some sort of integration in terms of what the actual habitat quality is across space, and in terms of how that influences survival downstream of migration and who actually returns. And so we look at the way of, measuring returns as a way to kind of integrate across some of the fact that salmon tend to like variable habitat conditions, and coming up with what the most suitable condition is for a particular population can be a complex question.
Are there things that are detrimental, like kind of sediment input from land clearing, and
yeah.
Are there things are bad for salmon, I guess?
Yeah, so copper is toxic.
Mm-hmm. Cool.
Copper is toxic, screws up their ability to actually home, or can be actually lethal. That would be a sublethal example. Sedimentation is hard because it gets stuck into the gills. They can't breathe as well. One of the biggest things that the Pacific Northwest region struggles with is the fact that they have had what were intact watersheds. They put dams and roads and culverts and basically death by 1,000 cuts in these areas where they used to be intact systems. So when you fragment the habitats, you reduce the connectivity, which I think we're showing here is an important piece of the puzzle in terms of reliable salmon production. So those are good examples.
Hey, So I'm wondering, you showed that some of the rivers in the watershed are glacially derived and some are not. And I wonder if annual variability in the amount of glacial input that some of those rivers are going might be able to explain some of these switches that you see in your data. Do you have any ideas of like how isotopes might potentially be applied to like get at some of those differential water sourcing questions, or do you think that's just not at all related to any of the?
Well, I think to the extent that it influences things like stream flow, which is a very stream flow and temperature in terms of how that plays out, especially during the growing season of fish, that's going to be an important piece of the puzzle. I think to figure out if it actually is driving some of this, we need more years of data across a range of climatic conditions. Right now, we've just got a few years of data, that are clustered together in terms of time. And so we don't really have the power, because we don't have contrast in the environment, and we don't have the number of years to kind of tease that apart. Okay. In terms of isotope approaches to that, I don't know. I don't know if it would be a better approach with just looking at, satellite data that are good proxies for temperature or stream flow or precipitation or snow cover, or coming up with some sort of sample-based, isotope inference in terms of, water sources or temperatures. I think it might be easier with the satellite type data in this region in particular. Yeah.
Cool. Thanks so much.